All right, let's dive right into this. Welcome back, Focus learners, to another explainer. Today, we are opening up a really fascinating masterclass on Chapter 3, Biomolecules. Basically, we're looking at the literal architecture of life. You know, we often look at the incredible complexity of the human body, or maybe a towering redwood tree, and it seems almost impossibly complicated. But when you really break it down, everything alive is built from a surprisingly simple set of building blocks. So today, we're going to see exactly how simple chemical monomers snap together to build the breathtakingly complex biological structures that make life possible. Here is our roadmap for today. We'll start by defining the general ingredients of life, and then we're going to break down the big four families of macromolecules. That's carbohydrates, lipids, proteins, and nucleic acids. And finally, we'll wrap the whole thing up with a quick summary. Okay, Section 1, the ingredients of life. From organelles to macromolecules, let's zoom in on what actually builds our cells. Moving right along to Section 2, carbohydrates, the energy source. Now, when you hear the word carbohydrates, you probably immediately think of food. And you're absolutely right, they are our primary energy source. But beyond just being an energy reservoir, they are incredibly important structural components, especially in things like plant and bacterial cell walls. To really understand them, we have to start with their smallest individual Lego brick, the monosaccharide. Mono meaning one, and saccharide meaning sugar. These are simple sugars that literally cannot be broken down or hydrolyzed into anything simpler. They're the absolute base monomer unit. Chemically speaking, they contain free aldehyde or ketone groups, and they follow a very neat, predictable general formula, CnH2ON. So for every carbon, there is a water molecule, which is actually where the name carbohydrate comes from. Pretty cool, right? And nature essentially categorizes these basic sugar bricks based on their functional groups into two main camps, aldoses and ketose. Aldoses have an aldehyde group. You'll definitely recognize glucose, which is our body's absolute favorite fuel. Ketoses, on the other hand, contain a ketone group. A great example is fructose, the sugar you'd find in honey or apples. So what happens when you start snapping these single monosaccharide Lego bricks together? Well, you get bigger structures. Snap 2 to 10 units together and you get oligosaccharides. Common table sugar, sucrose, is just 2 units joined together. When you string hundreds or even thousands of them together, you get polysaccharides like starch or cellulose. And importantly, they snap together using a very specific type of chemical glue called a glycosidic bond. Carbs can even link up with proteins or lipids to form glycoconjugates, which are absolutely crucial for cells communicating with each other. Next up, Section 3, lipids. Think barriers and storage. Let's shift gears from water-loving carbs to water-fearing lipids. Lipids are famous for being hydrophobic. They are nonpolar organic compounds that are soluble only in organic solvents, not in aqueous solutions. And that hydrophobic nature makes them the perfect building material for creating barriers, like the membranes that protect every single cell in your body. The key structural ingredient here is the fatty acid. Picture a long, zigzagging tail of a hydrocarbon chain with a carboxylic acid group tacked onto the very end. What's super fascinating from a biological synthesis perspective is that naturally occurring fatty acids are generally built up from 2 carbon units. Because they're assembled 2 carbons at a time, nature almost always produces fatty acids with an even number of carbon atoms. To build a full fat molecule, like a triglyceride, these fatty acid tails connect to a glycerol backbone using a specific bond called an ester linkage. Now, if you look closely at a fatty acid structure, like stearic acid, notice how biochemists actually number the carbons in the chain. They start right at the top with the carboxyl carbon, that's carbon number 1. Then they just count all the way down the hydrocarbon tail, ending at the methyl carbon, which for stearic acid is carbon number 18. But the shape and function of these tails depend entirely on one thing: double bonds. Saturated fatty acids, like that stearic acid we just mentioned, have 0 double bonds. They are entirely saturated with hydrogen, making their tails straight. This means they can pack tightly together and form solid fats at room temperature. Think of a stick of butter. Unsaturated fatty acids, however, have 1 or more double bonds, and that puts kinks in the tail, keeping them fluid at room temperature, kind of like olive oil. Oleic acid has 1 double bond, making it monounsaturated. Polyunsaturated acids have multiple double bonds. A really great memory hook here is arachidonic acid. It contains 4 double bonds, making it highly kinked and incredibly important for cellular signaling. Let's move to Section 4, proteins, the cellular machines. If a cell is a bustling city, proteins are the workers, the vehicles, and the machinery. They do almost everything. They act as catalysts in the form of enzymes. They are the antibodies in your immune system, the hormones sending signals, and the literal structural muscle fibers allowing you to move right now. Their phenomenal diversity comes down to just 20 naturally occurring amino acids. Take glycine, for example, to understand their anatomy. Right in the center, you have the alpha carbon. It's a chiral or asymmetric carbon attached to 4 different things: an acidic carboxylic group, a basic amino group, a single hydrogen atom, and lastly, the R group or side chain. That R group is the magic variable. It's the only thing that changes across the 20 different amino acids, giving each one its own unique chemical personality. So how do we get from single amino acids to massive complex proteins? Well, they link up. The amino group of one amino acid reacts with the carboxyl group of another, and when they do this, they release a molecule of water. It's kind of like a little water tax for building the chain. The resulting covalent bond is called a peptide bond. This creates a long linear polypeptide chain, which is really just the very beginning of a protein's life. Because a straight string of amino acids isn't a machine yet, it needs to fold into a specific shape. There are 4 progressive steps to protein structure. Step 1 is primary, that simple linear chain we just discussed. Step 2 is secondary. The chain starts to coil into alpha helices or fold into beta-pleated sheets, stabilized by hydrogen bonds. Step 3 is tertiary. The entire chain folds into a complex, fully functional 3-dimensional arrangement. And step 4, quaternary, happens when multiple folded protein subunits come together to form one giant supercomplex. It is literally molecular origami on an epic scale. Moving to section 5, nucleic acids, the information hub. So, if proteins are the machinery of the cell, nucleic acids are the instruction manuals. They serve as the core genetic and informational material for almost all organisms on Earth. And just like the others, they have a monomer building block, the nucleotide. A nucleotide has 3 distinct parts. First, a phosphate group. Second, a sugar. And third, a nitrogenous base. There are 5 possible nitrogenous bases in nature: adenine, guanine, cytosine, thymine, and uracil. The specific sequence of these bases is quite literally the code of life. The crucial point here is understanding the difference between the 2 main types of nucleic acids, DNA and RNA. Structurally, they differ in 2 major ways. First is the sugar. DNA contains a deoxyribose sugar, while RNA contains a ribose sugar. Second is the base substitution. While both use adenine, cytosine, and guanine, DNA exclusively uses the base thymine. RNA, on the other hand, swaps out thymine and uses the base uracil instead. Section 6, Summary of Building Blocks. Let's tie it all together. We have covered a lot of molecular ground today, so here is your ultimate cheat sheet mapping the 4 macromolecule families to their specific building blocks and the unique chemical glue that holds them together. Carbohydrates are built from monosaccharides linked by glycosidic bonds. Lipids are largely built from fatty acids and glycerol, snapped together with ester bonds. Proteins are chains of amino acids bound by peptide bonds. And finally, nucleic acids are polymers of nucleotides linked by phosphodiester bonds. Before we wrap up, let's put your focus to the test. Here is a quick pop quiz adapted directly from Chapter 3's review questions. We know the backbone of a single DNA strand is held together by phosphodiester bonds, but how do the 2 separate strands in a DNA double helix actually connect to each other, right in the middle? Is it A, a covalent bond, B, a hydrogen bond, C, a glycosidic bond, or D, a phosphodiester bond? Think back to our discussion on secondary structures and folding. I'm going to leave you with that provocative question to chew on. Finding the answer will absolutely solidify your understanding of how biology maintains its incredible, complex architecture. Thanks so much for joining me on this explainer. Keep exploring and stay curious.